Hello everyone, this is the continuation of third lecture colorectal carcinoma, and we are going to study molecular genetic pathways of colorectal carcinogenesis. So as we all know, there are two molecular pathways, adenocarcinoma sequence and microsatellite instability. In microsatellite instability, the clinical phenotype is hereditary non-polyposis colorectal cancer, and there is germline inactivation of MLH1 or MSH2 DNA repair genes, leading to mucinous and poorly differentiated carcinomas with lymphocytic infiltrates. The sporadic colorectal carcinomas are right-sided predominant carcinomas, and there is somatic inactivation of MLH1 or MSH2 DNA repair genes. And in adenocarcinoma sequence, the clinical phenotype is familial adenomatous polyposis, and the carcinoma are moderately differentiated adenocarcinomas due to germline APC inactivation. And sporadic colorectal carcinomas are left-sided carcinomas. Histopathology include tubular villus, and villus adenomas, and moderately differentiated adenocarcinoma. So this is the diagram showing how the normal colon is evoluted into adenomas and further into carcinomas. So on extreme left side, this is normal colon, which further lead to hyperplasia of colon, adenomas and carcinomas due to molecular alterations. So the main alteration is APC gene mutation, germline mutation or somatic mutation of cancer suppressor gene APC at chromosome number 5. APC gene mutation will lead to translocation of beta catenin. Translocation of beta catenin to the nucleus, where it activates the transcription of other genes, mainly MYC gene and cyclin D1, both of which are cell proliferation stimulators. So, mutation in APC gene will lead to translocation of beta catenin, which will activate transcription of genes like MYC and cyclin D1 and will lead to cell proliferation. Then mutation in proto-oncogene KRAS and loss of additional cancer suppressor genes like p53, loss of p53 and mutation in telomerase will lead to carcinomas. First adenomas, then carcinomas. So the adenocarcinoma sequence leading to carcinomas. The evolution of carcinomas from adenomas, from hyperplasia, from normal colon. So normal colon is transformed into carcinoma via adenomas. So basically the gene involved are APC gene. These are tumor suppressor genes: APC gene, KRAS proto-oncogenes, P53 and telomerase. So this diagram is showing the microsatellite instability pathway. Microsatellite instability. In microsatellite instability, basically there is mutation in DNA repair genes like TGF beta, BAX. TGF beta receptor gene normally inhibits cell proliferation, but in mutated form, it allows the uncontrolled proliferation of colonic epithelium in adenomas, leading to carcinomas. The BAX gene normally causes apoptosis, but a defect in it will result in loss of apoptosis and dysregulated growth, so leading to carcinomas. TGF beta, normally it is a inhibitor of cell proliferation. So mutation in this gene will lead to uncontrolled cell proliferation, and mutation in BAX will lead to inhibition of apoptosis, so further leading to dysregulated growth. So this slide is showing the summary of molecular defect in colorectal carcinoma. So there is mutation of APC gene at chromosome 5. This gene regulates level of beta catenin. There is point mutation of KRAS oncogene. There is deletion of suppressor gene on chromosome 18, loss of P53 and activation of telomerase. This all gene mutation is seen in adenoma carcinoma sequence or pathway. And the microsatellite instability pathway, other pathway characterized by genetic lesions in DNA mismatch repair gene. It is seen in 10 to 15% cases, especially in HNPCC syndromes. Thank you.